Okay.
Morning, everyone. This morning we will be here for holding the master degree defense. Therefore, students will take part in the defense, and their thesis have passed the review, and their credit hours finish their credit hours, and they satisfy the defense condition. Today, we invite the President, Professor Wang Zhenshan, from Hebei University, and other committee is Professor Ge Shujun, Professor Chang Jinhua, Professor Yao Zhanjun, and Professor Duan Huijun. Okay, next we will let the President for our defense. Okay, welcome.
Okay, let's start. Everyone, every student, the final report no over than 20 minutes. The answer question time is around 15 minutes. Now, you start first.
Okay. Good morning, everybody. Good morning, distinguished professors. Thank you, Mr. Chairman, to give me this podium today for my presentation. So I think the time is very tough. As much as possible, I will try to manage. Maybe. Thank you. Now I will continue for my presentation. So if there is any unclarity, you can interfere me, so I can continue. This is my research topic, genetic variability and QTL mapping of food and seed-related traits in cultivated peanuts. This is my outline. I have introduction, significance and objectives of the research, materials and methods, result and analysis, and conclusion. Finally, I will have acknowledgements. So let's go from introduction. As you know, peanut is one of the important oil and legume crops. It grows more than 108 nations or countries in the world. So peanut is now this time the sixth important crop in the world. So the world average productivity is 1.6 ton. So one of the leading peanut producer countries, China, is the biggest producer and leading peanut grower country. So this is the world productivity amount now this time. Peanuts grown extends to more than 100 nations. It is already obvious. When we look at the nutritional content of peanut has high oil. The peanut seed, approximately it has 50% of oil, 25 up to 30% of protein, and the remaining percent is others. Now, my research is focused on the peanut molecular breeding. So I have used the AFLP and the SSR markers to screening out those real populations. So this is the genomic acid breeding have the following improvement strategies. One is the marker acid backcrossing. The second is marker acid recurrent selection. The third is genomic selection. As you see from this slide, this is a schematic diagram of the molecular breeding and marker acid selection in peanut, as much as possible in other crops. That is all for my introduction. Now come to my objective. The present study has the following objectives. Objective 1, to estimate genetic variability, heritability, genetic advance, and breeding values for each real population. The second objective, to screen out outstanding and promising transgressive real populations for advanced breeding programs. The third objective is to construct genetic linkage map for cultivated peanut. The fourth objective is to mapping pod and seed related quantitative traits. So when you come to materials and methods, initially I have phenotyping. Those real populations constructed by peanut team members. So the present study was carried out using 251 real populations. The field experiment is designed using RCBD design without any replication. It conducted in 2 locations, Boding and Handan, for 2 consecutive cropping seasons. This is a field experimental site in Boding, Xinhuan. Now come to the estimation of breeding values and index selection. To calculate the estimation of breeding values, we shall use best linear unbiased prediction, or BLAPS or BLUE, computed for calculating narrow sense and broad sense heritability to estimate the breeding values of peanut. So BLUEs obtained with different estimates of narrow sense heritability were highly correlated with regression determination values. Breeding values predicted by BLUEs were correlated with line means that are estimated from a fixed effect models. I have used fixed and random effect models by using systematic statistical analysis software, or SAS. So the BLUEs research approach has used in this study was the only procedure proven effectively to predict biparental single cross populations to screen out the best outstanding lines. So now we come to the genotyping and mapping populations. As I mentioned earlier, I have used 251 real populations to construct genotyping by using the SSR and AFLP molecular markers. So mapping populations are the prerequisites for genotyping and mapping populations to study the genetic factors, structures, and the organization of the genome structures of one organism. This is the parental, the 2 parents. The crossing is made from P1, female parent Silihong, and P2, male parent, we call black peanut. You can see the seed color and the other seed morphological characteristics. Okay, this is the seed of real populations. Yes, this is the polymorphic primer screening. Gene electrophoresis was conducted to screen out the most high polymorphic molecular markers from the parental populations, from P1 and P2. Yes, this one is a crossing block. We have made a cross from this crossing block. So this is a crossing block nearby our school. Yes, I have used this primer combinations. M.Sc and Ecorai primers. This is the list of primers what I have used for screening. This one is also the primer. Yes. all 251 repopulations with 2 parents were planted in peanut germplasm resource laboratory for DNA extraction. After 15 to 20 days of planting, I have extracted the DNA from 251 repopulations and from 2 parents. Yes, this is the germination or the population is growing in the laboratory for DNA extraction. Genome DNA was extracted by using the CTAB modified protocol methods. As you know, AFLP molecular marker needs a high genomic and high quality DNA for conducting polyacrylamide gel electrophoresis. Yes, this is the DNA extraction procedures. This is the DNA extraction. I have followed this step to finalize the whole FLP molecular marker analysis. The first step is digestion of the genomic DNA. I have used the incision enzyme to cut the genomic DNA, to cut the double-stranded DNA to single-stranded DNA before conducting the adapter ligation. So the second step is to ligate the adapters at both 5' and 3' ends. The third step is pre-amplification. When we have to pre-amplification, our band must be smeared. No need of band for that time. Just this step is a prerequisite for amplification procedures. The fourth step is amplification. And the fifth step is polyacrylamide gel electrophoresis. And finally, scoring DNA fragments or bands from that gel. Then finally, I have doing data analysis by using join maps and other softwares to make a genetic linkage map. This is the whole procedure of AFLP molecular marker procedures. Yes, this is a diagram what look like the AFLP molecular marker. The whole steps. Yes, this is a positional cloning of pages to making the electrophoresis. When you come to data analysis, code and serial traits were subjected to system analysis software or SAS by using the DMRT and LSD. That means Duncan's multiple range test to screen out the most outstanding repopulations from the whole population. The data normally was tested by using proc univariate before conducting the data analysis to check up the data normality. And finally, I have used the Shapiro-Wilk statistics to check up the null hypothesis. Correlation coefficient among pod and serial traits were estimated using the proc-GLM and proc correlation models by using system analysis software or SAS. And also proc ANOVA I have used it for making the ANOVA analysis or the ANOVA table. And finally, I have used the LSD or least significant difference to know the difference between those three populations. Okay, I have used this model to calculate the ANOVA procedures. This is the mathematical models to conduct the ANOVA. Yes, this is the physical skeleton of the ANOVA table. Phenotypic and genotyping variability. Expected mean squares under the assumption of random and mixed model was calculated from a linear combination of phenotypic and genotypic coefficient of variation, which were computed as per the method suggested by Levin. This is a mathematical model to conduct the genetic and phenotypic variability of the populations to estimate the genotypic and environmental variances and also phenotypic variances. I have used this mathematical equation. Yes, both genotypic and phenotypic coefficient of variability were computed as per the method suggested by Burton and Devin. As you see, this is a mathematical model, the genetic coefficient of variation, how to calculate. So I have used this model to calculate the genetic coefficient of variation, and also I have used this model to calculate the phenotypic coefficient of variations between real populations and parents. Okay, this one is a heritability analysis. To calculate the broad sense heritability, as you know, heritability is the most important to screen out the real populations. Is it heritable or not? To select our populations, we must be now the heritability value of those populations, both broad sense heritability and narrow sense heritability. So this is a mathematical model how to calculate the broad sense heritability. To select our best genotypes, our heritability value must be greater than and equal to 60%. When our value less than this percent, our selection will not be succeed. Yes, this is a mathematical model for calculating the narrow sense heritability. Yes, I have used the regression analysis to calculate the narrow sense heritability and to compare and contrast both narrow sense and broad sense heritability. How the additive gene effect and how the dominant gene effect and how the environmental effect control our populations. So I have used this model to estimate the difference between the two heritability, broad sense and narrow sense heritability. Yes, this is a genetic advance. How much percent we gain the gene from the parents, from the female and male parents. So to know that the genetic gain, we must be calculate the genetic advance values. So to calculate that genetic advance, we have used heritability and selection intensity, and also we have used the standard deviations from the phenotypic data. Yes, this is the genetic advance as percent of mean. To select the high genetic gain, so our genetic advance value must be greater than and equal to 20%. When our value less than this percent, the genetic gain is very poor. So we cannot confirm the genetic gains from the parents. The correlation analysis. As I mentioned earlier, I have used the pro-correlation statistical models to calculate the genotypic and phenotypic correlation values. Is it correlate directly or indirectly? This is a mathematical equation to calculate the correlation analysis, both phenotypic and genotypic correlation values. So this is a genotypic data analysis. To make a genotypic data analysis, I have used several softwares. I have used join map to construct a linkage map, and I have used IC mapping also, and I have used plant building tools. I have used QTL cartographer, and I have used QTL network. I have used QTL map makers to design and to know where is the position of the QTLs. Yes, I have used this join map software to make a linkage map with a LOD value of 4. The marker orders were confirmed by comparing the log likelihood of the possible orders using the multi-point analysis. QTL mapping. The present study QTL mapping on pod and cell relate rate were calculated using the phenotypic mean separation values. So QTL analysis was computed using the composite interval mapping in the QTL cartographer software. Result in analysis. Based on system analysis software in the model of ProGLM, variance estimation analysis and ethical statistics results, the null hypothesis was rejected due to significant mean difference between real populations and environmental variations in the traits of port and seed-related traits. The trait of port length showed non-significance between treatment. That means the F-test of the null hypothesis was accepted. When you look at the traits of port width, port thickness, port length by width, thickness of port shells, and multi-port seeds per plant showed there was a significant difference between treatments, between environments and within seasons. This is the ANOVA table. For seed-related traits, analysis of variance showed that were a mean difference between treatments and environmental variance at 0.05% level of significance. Yes, this is ANOVA table of seed traits. The previous one is the ANOVA table of, this one is the ANOVA table of the port traits, and this one is the ANOVA table of seed traits. The comparison of port and seed relates across two environments. In Handan, port length by width and port length traits show less relative determination correlation values, but the remaining traits have expressed strong correlations. In porting, the traits of port length, seed length, and seed thickness had expressed perfect correlation determination. When we call perfect correlation, our correlation must be greater than 96%. So this is the descriptive statistics of this correlation analysis and the whole two environmental boarding and Handan. This is for boarding. This one is mean separation of P1 and P2, and this one is for real populations. This one is for Handan. Yes, the Pearson correlation analysis, genotype by environment, combined Pearson correlation coefficient values between -1 and +1 indicated that majority of the correlation matrix between port and seed-related traits had a positive correlations. Port width traits was positively associated with port thickness, seed length, seed width, seed thickness, thickness of port shells, and so on. This is the correlation tables. In general, the correlation between port and seed-related traits across the two environments. This is the GBA combining of two environments, boarding and Handan, in 2 seasons. Is the AMI biplot estimation. I have used the AMI analysis of additive molecular variance analysis. I have used the biplot estimation of two environments, boarding and Handan. So AMI model combines the analysis of variance for main effect of genotype plus environment with the principal component analysis of genotype by environment interaction. So AMI biplot has proven to understand the complex genotype environmental interaction effects. So in AMI biplot, I have estimated, I have selected, I have screened 4 best genotypes for boarding. This is environment. Environment 1 means boarding. Environment 2 means Handan. So I have selected the best 4 genotypes for boarding. Genotype 122, 128, 58, and 227 for boarding. So I have screened 4 genotypes for Handan, as you can see from this. This genotype or the population distribution in the two environment. This is the small dots indicated. This one is environment 1, this side is environment 2 or Handan. So this one is the red dot is indicated the real population is. So the distribution of real population is per each environment. So it looks like this one for port length traits and port width traits. For seed traits, we can see the distribution of those real populations in the two environments. Yes, this is the Hebei combined discrete statistical analysis for pod and seed related traits. Yes, I have used this Shaprowick statistics to estimate the significant difference between those real populations and parentheses. I have conducted the frequency distribution analysis. So the frequency distribution of pod and seed traits across two environments within two consecutive cropping seasons was computed in the model of PROC mixed SAS models. Based on this model, I have put the random and fixed effect models. So this is the frequency distribution of those real populations and their traits. So as you can see, I have used the Riemann models, restricted maximum likelihood models, to estimate the residual and the frequency distribution of pod and seed traits in the two environments combination. This is for pod traits and this is for seed traits. The genetic variability and heritability. Genetic advance measures the expected genetic progress that would be resulted from select best performing genotypes based on a given traits. So the genetic gain that can be obtained from a particular trait through selection is the product of its heritability, phenotypic standard deviations, and selection differential. Yes, this is the most important table for my research. You can see this is the genetic variance, environmental variance, phenotypic variance, broad sense heritability, genotypic coefficient of variation, phenotypic coefficient of variation, genetic advance as percent of mean. The highlighted part, this part, as you can see, the heritability value is very high. That means the crossing is successful. So we can select confidentially our real populations. So in this part, you can see the heritability value both. Let me. This part, we can see the heritability value between broad sense and narrow sense. We have equal value. This means no environmental effect, no any dominant gene effect. This indicates the additive gene effect is very high. So we can confidently estimate and select those genotypes cannot affected by any environmental variations or environmental changes. We can confirm by conducting, by look at this value. You can see this one is the additive gene variance. This one is the additive gene variance by environmental interaction. This one is the dominant gene variance. This one is the dominant by environment interaction. This one is narrow sense heritability, broad sense heritability, and dominance ratio. So based on this table, sorry about this one. Based on this table, we can estimate our real populations. We can select confidently. So this is a heritability value without any restriction maximum likelihood values. But in this part, we can look at the genetic gains. As I told you earlier, our genetic gain value must be greater than and equal to 20%. So in this table, you can see the highlighted part. High genetic gain. We have a high genetic gain. We have a high genetic gain. The highest genotypic coefficient of variability had been observed in multi-seed pods. Pod weight, number of pods per plant, and pod length, seed weight had a moderate value. The remaining traits have shown no genetic variability. The highest phenotypic coefficient of variation, as you know from here, had been recorded in the traits of pod weight per plant, multi-seed pods per plant, and so on. Therefore, the present study promising